So now we move forward to link reaction. We got pyruvate from glucose during glycolysis, and now these pyruvate enters into the mitochondria. So these reactions are actually happening in mitochondria. These reactions are happening in mitochondria. Pyruvate is oxidized to acetyl-CoA. You have to remember that pyruvate contains 3 carbon while acetyl-CoA has 2 carbons. So the 1 carbon is used in making carbon dioxide. Right? The enzyme is dehydrogenase, so NAD must have to be reduced to NADH, and this is link reaction. Now the acetyl-CoA, which is produced in the link reaction, it goes to citric acid cycle, which is also called tricarboxylic acid cycle or Krebs cycle. And this acetyl-CoA can also be used to make fatty acids. We'll see how. Now a few words about galactose metabolism. So we get galactose in our diet through lactose. Lactose is hydrolyzed or digested by an enzyme lactase to make glucose and galactose. You know glucose is going to make pyruvate. Now this galactose is absorbed into the blood, and from the blood it goes to the cells. Again, this first is trapped by the enzyme galactokinase, and we get galactose 1-phosphate. Then that galactose 1-phosphate makes glucose 1-phosphate, and then this glucose 1-phosphate makes glucose 6-phosphate, and then this glucose phosphate move into the glycolysis to make pyruvate. So actually galactose also makes pyruvate by this process. And just an important point, and this is really very important, very important, that if there is high level of galactose in the blood because of galactokinase deficiency or by galactose 1-phosphate uridyl transferase deficiency, galactose accumulates in the blood, and that is called galactosemia. And if there is high level of galactose in the blood, this galactose enters into the lens, and inside the lens there is an enzyme which is called aldose reductase, and that aldose reductase reduce this galactose to galactitol, and this galactitol exerts osmotic pressure, and because of that osmotic pressure there is swelling and cataracts in the patient who has this galactosemia. And this actually is also the basis of retinopathy and diabetes. If we have high level of glucose in the blood, that glucose enters into the lens, and the same enzyme aldose reductase, because glucose is also an aldose like galactose, aldose reductase converts glucose into sorbitol, and sorbitol exerts osmotic pressure, and because of that osmotic pressure there is swelling in the lens and the cataract formation. So that is the basis of retinopathy and diabetics. So now few words about fructose metabolism. And in the fructose, again fructose, we get fructose from sucrose, and sucrose is hydrolyzed to make glucose and fructose. Glucose follows the glycolysis, and fructose again is first phosphorylated to fructose 1-phosphate, and then this fructose 1-phosphate, here the enzyme is aldolase B instead of aldolase A, that is used in glycolysis. This fructose 1-phosphate is cleaved to dihydroxyacetone phosphate and glyceraldehyde. This dihydroxyacetone phosphate then can go to the glycolysis or glycogenesis and gluconeogenesis, we will see. And this glyceraldehyde can be converted to glyceraldehyde 3-phosphate, and then this glyceraldehyde 3-phosphate makes glycolysis. So in effect, either this is galactose or this is fructose, they actually join in glycolysis to make pyruvate. Alright.